All right, so in this pre-lecture, we're going to talk about the different types of disinfection that you can potentially use in drinking water treatment. So we're going to talk very broadly about disinfection, and then we'll talk a little bit more about specific types when we're in class. So what is it? What is disinfection? So it's the inactivation or the destruction of pathogenic organisms in water. So this is different from what we call sterilization. So sterilization is defined on the bottom of this slide. Sterilization is the destruction of all living organisms, especially microorganisms in a system. It's not just targeted organisms. Disinfection is targeted. So it's meant to remove specifically the organisms that are going to cause disease, so the pathogenic microorganisms. So when we talk about disinfections, there's two different ways of doing it. There's what we call destruction, and there's what we call inactivation. So destruction is going to kill the organism by either lysing the cell or disrupting metabolism to cause cell death. So lysing the cell means the chemical comes into the cell and it breaks it open. And then disrupting metabolism, disrupting chemical processes in the cell can eventually lead to cell death as well. So generally, destruction happens through chemical disinfection. So we apply something like chlorine or ozone. And then inactivation is different. This is a different process where we focus on specifically rendering the organism unable to reproduce. So many times this is a physical disinfection process, and we use ultraviolet light. So two different ways of disinfecting, destruction and inactivation. And the purpose of disinfection is very straightforward. It's to prevent waterborne illness caused by pathogenic organisms, right? So we've gotten rid of all of the other stuff in water by the time we get to disinfection. And at the end of the day, all of our regulations, all of our guidelines are all about human health and safety. So disinfection is the key barrier in pretty much any drinking water system. You're going to need disinfection unless your source is very, very clean. So it's normally the last step before the water goes out to the distribution system. So how does it work? We briefly talked about this. Chemical disinfection works with, as its name implies, a chemical is applied to the cell, and it's going to poison the cell, right? Basically, it's going to disrupt metabolism, it's going to disrupt chemical processes in the cell, and it potentially lyses the cell and causes cell death. So you're killing things off with a very, very stringent chemical. Physical disinfection is when we apply a process that destroys or damages cell DNA most of the time. So what's going to happen with a physical process is instead of applying a chemical, we apply a stimulus like a radiation, ionization, and ultraviolet light. And it's going to render the cell incapable of reproducing. So it destroys components of DNA so that DNA cannot replicate when a cell undergoes meiosis and mitosis. So the cells can no longer reproduce and they're not able to create more, right? Because we need an infectious dose when we have a waterborne disease. So you need enough of the organism. And so this keeps the population of organisms down in the system, and it does kill some of them as well. So, key concepts when it comes to disinfection. So, disinfection is a rate process. So we talked about kinetics at the beginning of this class. Chemical disinfection in particular is governed by kinetics, as well as some of the physical disinfection processes that we have. So because it's a complex rate process, it depends on the physiochemical nature of the disinfectant. So what type of disinfectant it is and how that disinfectant reacts in water. What are the biological characteristics of the pathogens or microorganisms that you're trying to disinfect and get rid of? And then the interaction between that disinfection and the pathogen. And this is related to what we call contact time. So just putting a chemical in the water is not going to immediately kill everything that's there. We need a certain amount of time for that chemical to react or that disinfectant to react in the system and have that contact time to actually basically come into contact with all of those microorganisms. And then disinfection is affected by several water quality parameters. It's affected by temperature, it's affected by pH, and pH is a big one when it comes to chlorine. It's affected by electrolytes, cations, anions in the water, and then other interfering substances. So a good example of this is organic matter, like natural organic matter. Chlorine likes to react with natural organic matter, and it likes to react with nitrogen compounds as well. And that is going to decrease the amount of chlorine that's actually effective for disinfection, which is something we'll talk about in class. So you can have other interfering substances that get in the way of disinfection. So when we actually design these things in the field, key components are the size of the basin and the geometry of the basin for contact time. So what you'll see here on the screen on the top and on the bottom are an example of baffled contact chambers for water. So the baffles in this system force the water on a kind of a serpentine path throughout the system. And what that does is it keeps the water in there longer so that there's a longer amount of time that the disinfectant has to have with that specific microorganism that you're trying to get rid of. So what you see in the middle on the right side is a very simple way of dosing chlorine, just having a contact tank, having chlorine chemical being pumped in, and then it sits in the tank and has contact time. So these systems can be very simple, but they can also be very big, and they can work at different scales. So typically in a bigger water treatment plant, we see those baffled reactors so that we have enough time because we're treating so much water. But in smaller systems, our contact tanks can be very, very small. The other thing we need is we need a device to apply the dose. So this is normally some kind of a chemical feed pump when it comes to things like chlorine and maybe ozone. But when it comes to UV, we need to make sure that we actually have the UV lights. So you can almost think of like a fluorescent tube, like in your lights overhead in a classroom. Same kind of idea with a UV light, but it's emitting light at that UV range instead of visible light, which is what you see from a fluorescent light. So on this slide, these are just some pictures of kind of how everything works. So let me grab my laser pointer. So in that top left corner, this is what's called cytolysis. So this is when the cell lyses and bursts open. So this is just demonstrating how, in this case, it's water. Water comes into the cell, it causes the cell to expand and the cell burst. The same thing can happen with a chemical. A chemical can come in and it can cause damage to the membrane of the cell and cause the cell to burst. And then down on the bottom is an example of a very, very simple chlorine system where you have an analyzer, a dosing pump, you have some sort of chemical, you have a pump, and then you have a potable water storage tank. So you have contact time. In the top right, you're going to see what UV does, and this is actually what UV does to your skin cells when you get a sunburn, but it does this inside of organisms as well. What will happen is UV will break bonds between these different bases in DNA, your A, C, G, and T, if you remember your biology, right? It's going to break those bonds, and it's going to create what we call dimers, and it's going to keep that DNA broken so that when the cell goes to replicate and it replicates its DNA, it no longer can because things are bonded incorrectly now. So that's how inactivation works. What you'll see in the middle is water going in, UV lights kind of on the edges of those tubes, and water coming out. So many of our UV reactors are cylindrical. And then unfortunately my face is in the way, but down below you have kind of the spectrum of light where UV is most effective. So UV is most effective in what we call the UVC range. So it's not visible light, right? It has a higher wavelength and a higher energy than visible light. All right, so a couple things engineers need to know when it comes to disinfection. You need to know chemical demand in or of your water. So many times this is chlorine demand. So we're going to have water quality parameters present, such as nitrogen, natural organic matter, and they're going to eat up chlorine faster than the chlorine can actually disinfect the pathogens. And so we actually have to apply a dose that accounts for those parameters, and we'll talk about this in class. We're also going to need to know the dose of the disinfectant. So it's based on chemical reactions and water quality. How much do we actually have to put in? And when it comes to, I don't have this on the slide, but when it comes to UV disinfection, you need a look at what we call transmittance. So how much UV can actually push through that water. So if you have very turbid waters, high turbidity levels, it's going to be harder to disinfect with UV. So that's something to keep in mind. We also need to know how many log removals are needed. So we'll talk about this on the next slide. And then it's also good to know what organisms need to be removed. Sometimes this is harder to figure out in systems than other, but for chemical disinfection, normally that will kill most bacteria and some protozoa. UV disinfection is pretty good at getting everybody in the water, so viruses, bacteria, protozoa, because it goes after DNA, as opposed to trying to kill the cell. So let's really talk really quickly about what we call log reductions. So log reductions are a way that we look at how we remove organisms from a system. So instead of having an MCL and a target that we can reach, that's not really feasible for microorganisms because it's hard to measure microorganisms in water. So we use what's called a log reduction scale. And so what this relates to is the percentage of microorganisms that are physically removed or inactivated by a given process. So you'll see physically removed is part of this, right? So we talked about how filters can actually remove organisms from a system, and that might be something like a 1 log reduction, meaning that a filter gets rid of 90% of the organisms that are in the system. So you'll see that this is what we call a log scale, right? So 1 log reduction means a 90% removal, 2 log reduction is a 99% removal, 3 log is 99.9% removal. And so depending on what you're trying to do in your system, if it's surface water or groundwater and what you're trying to remove, you need specific log removals. So for example, surface water treatment requires what we call a 3 log removal for Giardia. So throughout your water treatment system, you must remove 3 log. You must show that you have 3 log removal. So your filter may give you 1, your sedimentation process may give you 1 log removal, and then you need an additional log removal to make that 3 via disinfection. So each organism has a different log removal that needs to be achieved so that we don't have that infectious dose anymore. So this is important when it comes to disinfection because disinfection is going to give you one of the biggest log reductions that you can have in a system. So some key calculations that engineers are going to need to know. We're going to talk about these in more detail in class, but you're going to need to know dose. So on the left side, we have an example chlorine dose calculation. We have an example calculation for UV dose. The Chicken-Watson law is a disinfection law. It's based on first order kinetics. So this is very similar to what we've seen before with kinetic reactions, and we'll talk about this in class. And we need to know contact time. This is really the key one that you need to be able to calculate. You need to understand the concentration of a disinfectant at a given time and the time of contact required to kill a specific organism. Those are going to be important for you as engineers. Last thing we'll talk about real quick with disinfection is some advantages and disadvantages of these particular oxidants, disinfectants. So chlorine you can have in terms of sodium hypochlorite or chlorine dioxide. The advantages of the chemistry is well studied. We know a lot about chlorine in the system. It's been used for years. There's wonderful graphs out there that show you that when we started using chlorine, typhoid cases went down by millions across the world. The disadvantages of chlorine is that it creates what are called DBPs, disinfection byproducts, and the fact that it's pH dependent. So whatever pH you have in the water has an impact on how potent that chemical disinfectant is. And we'll talk about DBPs on the next slide. Ozone has the advantage of not creating those DBPs. So DBP again is disinfection byproduct. It is a very strong oxidant because it's O3, so it can also oxidize other parameters. So it's actually used sometimes to get rid of manganese and iron. The disadvantages are that it normally requires a little bit higher chemical dose, so you go through chemical faster with ozone sometimes, and it needs more specialized equipment because you have to have ionizing reactions in your system. Ultraviolet has the advantage of removing protozoa that are resistant to chlorine, but they have the disadvantage that they sometimes need a constant and consistent power source. So if you're in very rural areas, sometimes these are hard to implement if you have power outages. So last thing on disinfection, some potential impacts. So we mentioned DBPs. So when we have a chemical disinfectant reacting with other things in the water, we create what are called disinfection byproducts. Some are hazardous and some are not. But the ones that we care about are what we call trihalomethanes, which are THMs, and haloacetic acids, HAAs. And so these are chlorinated disinfection byproducts, and they are carcinogenic. So they cause cancer, and they are regulated in the United States. So we have to be careful with the dosing that we have in the system, and we have to remove all of our natural organic matter and our particulates ahead of disinfection. So these trihalomethanes and haloacetic acids are formed when natural organic matter reacts with chlorine. So we have to have good filtration and sedimentation ahead of disinfection to avoid these. And then finally, when it comes to disinfection, we have chemical costs many times associated with these processes, right? So you have to buy chlorine or ozone, and you have to have power for UV. So there's cost associated with maintaining these systems. And then finally, if you have chemicals, you now have to worry about chemical safety. So you have to be able to store these chemicals in a safe manner and apply these chemicals in a safe manner. So those are some things to think about when it comes to disinfection. So a little longer pre-lecture today, and I apologize for the length of it, but I hope that gives you a good introduction to disinfection. We will talk more specifically about each type of disinfection and some calculations that you can do for disinfection when we talk in class.